All right, let's get started. We'll do the best we can with what we got. Let's see. Yeah. All right, so starting off with the cell. Can you see me? Do I need to be on the...
Turn off the light.
Turn off the light? Okay, I'll turn off the light. Turn off the lights. I don't know where the light switch is. Oh, it says, please do not turn off the lights in this room. Ha! Well, we got to do the best we can. That's what it says. All right. Yeah, it probably connects to something over there. It is what it is. We'll just have to do. We'll work with what we got. All right. So first of all, let's continue to talk about biology and what makes up the different plants and different animals and everything that we're studying, and what the building block of life is known as the cell. The cell, the smallest unit of life that still has the characteristics of that life, right? So we, as human beings, we all started off as single cells. You getting this?
No.
Smallest unit of life that still has all the characteristics of that life. Cell, right? That's what a cell. We all started off as cells. Sperm, egg, fertilized. The sperm fertilized the egg. It was a single cell that eventually migrated into your mother's uterus and grew, went through the process of mitosis, multiplied many times, and became human beings, which you are now, as millions of cells that make up the human being, right? Plants also made of cells. Okay, smallest unit of life is the cell. Cells weren't discovered until microscopes were invented. We were able to look at something on a microscopic level, see this. We were able to, cells were what made up living beings, living entities. So cell theory was developed in the mid 1800s. We had microscopes that could look at cells. Early cell theory said all organisms are made of one or more cells. The cell is fundamental unit of life. All cells come from pre-existing cells. Cells don't come from nowhere, all right? Cells came from other cells, right? Cells have the same basic chemical composition. We see the nucleus, we see the organelles, we see the cellular membrane or the cellular wall, which you'll see. Cells use energy. Remember energy as one of the aspects that helps us to recognize that something is alive. Cells contain DNA. DNA is duplicated to make new cells, right? So whenever we started, like I said, we all started off as one cell, and then it became 2 and 4 and 6 and 8 and kept multiplying. The DNA in your original cell, which is half the DNA from your mother, half the DNA from your father, made the complete set of DNA that is what you have in your body right now. And then that DNA is copied each time your cells are copied. Okay?
23 or 24?
Each makes it 23, makes a set of 46. Correct.
And when you
Cell. Go ahead. No. Well, you came from the egg in your mother and the sperm in your father. That made you.
You said cells come from pre-existence.
So saying a new cell, yeah, and it's right,
okay.
Exactly.
Okay.
There it is. It's clicking. All right. Most cells are too small to see without a microscope. With the naked eye, they're too small to see. So we need microscopes, which we're going to use. If you've seen in our lab room, we have a whole cabinet full of microscopes, which we haven't even touched yet. And so we're going to use those microscopes. I'm going to teach you how to use those so that you can see things like cells. Yes, question.
It said most cells are too small to see without a microscope. Which cells are big enough to see without a microscope? Like it said most cells. Oh.
Frog eggs. All right. But yeah, generally you cannot see a cell without a microscope. And you need a high, depending on, you can see here, range of the human eye, range of a light microscope, and then range of electron microscope. Ant is not a cell, just in case you were curious. All right, so we have different types of microscopes to be able to see the cells, right? We're gonna use microscopes in lab, and so you'll be able to see this for yourself. A transmission electron microscope, a very powerful tool for seeing the internal cell structure. So not only can we see the cell, which is microscopic, you can't see it with your eye, but we have the technology to be able to see inside the cell, right? Which helped us to understand the organelles and the different parts of the cell.
That's what that is? Helps us see inside of the cell?
Yeah, the TEM, transmission electron microscope, is a microscope that can help you see inside the cell. And we can see what the cell is made of, right? So down to small molecules and atoms, proteins that are inside of the cell. Okay? SEM, scanning electron microscope, also very powerful, can reveal details on the cell surface. That's also something. Oh, go ahead, question.
So there are multiple types of microscopes?
Yeah, there's different types of microscopes.
So,
There's
the basic kind of
the basic one that we're gonna use is the one in the lab. The
can only see
the one that we're using can see up to 100 times magnification.
so the whole cell or just parts of it? 'Cause I know
No, we can see the cell.
you see parts of it. Is that
You can see a little bit. You can see the fact that there is something in there, but it's hard to see exactly what it is. Yeah.
advanced microscopes that can allow to see different parts of the cell?
You talking about in the lab?
Yeah, is that you just have a basic microscope?
I mean, basic is pretty strong. I mean, to be able to magnify something 100 times is pretty strong. But no, but we don't have. You're not going to be able to use. I mean, this is like $1 million piece of equipment, not million, but like couple $100,000 piece of equipment. We don't have those available for the students to use. No. Not to say that you're not worth it. It's just that we don't. It would cost a lot of money to give every student one to be able to use. All right, but yes, but we can look at the pictures that it makes. All right. This is the microscope that we will use. Light microscope. Microscopy allows us to view cells. So this is what you would see. See, you can see the cell, and then you can see that there are some things inside of it. Now, it's difficult to distinguish, but we're still able to, as you can see, the range of light of the light microscope. We are still able to see plant and animal cells, and even bacteria, and right about almost to the size of a virus. We can see with that. So that's what we have in the lab. Okay? So no, we're not going to get all the way down to proteins and atoms and small molecules. We don't get that small with what we're able to see, but we're able to see quite a bit. You need to see that for purposes of the class. All right, so cells vary in size. You have different types of cells. Different sizes of cells. So bacteria, archaea cells are 10 times smaller in diameter than most plant and animal cells. Not all cells are equal size. Also, cells are not the same shape. Not all cells are. Some of them with the microscope. Some of them, viruses, you need an electron microscope. So just depending on the size of the cell. All right, this is also talking about the size of the cell. Thousands of bacteria cells would be able to fit inside one human cell. Anybody ever had a bacterial infection? Yes, and we all have, right? And so the bacteria can infect the cell. It can go inside of the cell. Okay, go ahead.
I've kind of probably a stupid question.
No, there's no stupid questions.
But How did the scientists know that that's what the inside of the cell looks like? How did they figure out that can they literally see that with the microscope?
This transmission electron microscope. They can see the inside of the cell. This is them looking at the inside of a cell. Yeah, and they studied those things. Yeah, you can see all the way, these microscopes, you can see all the way down to the size of an atom.
If you put your hand under a microscope and got it really, really small, would you see atoms?
So they take really thin, because they have to shoot the light all the way through it,
Oh, okay.
right? So you need a real, so you'd probably take like a little bit, you could probably take some cells off of the surface of your skin and then put it under, on a glass slide and then shine the light through it and be able to see it. All right?
Question.
Yeah.
How can you take cells off the surface of your skin?
What do you think's on the surface of your skin?
The cells.
So your skin is made up of, the surface layer of your skin is called epithelium, right? So do you go get your nails and toes done? Oh, you don't do that? Okay, sorry, bad example. So, for instance,
Do you exfoliate? Do you do exfoliation on your skin?
so when you exfoliate, you're actually removing dead skin. That's the top layer of your skin. And so if you were to commit a crime, which I'm not saying you would ever do such a thing, but if you were to, this is why it's so difficult to get away with things these days when they use the technology that they have available to them. But if you were to commit a crime, all I need is for you to have rubbed up against something and some of your skin to have been left, when I touch something, you can't see it, but some of my skin, my dead skin is left on this surface. So that's why they're scanning these surfaces and all they need is a little bit, because if they can find a couple cells of dead skin, they can find my DNA. Because all of my DNA is the same DNA that's in the dead skin cells is the same DNA that's in the rest of my body. And if you find my DNA, then you found me. Did I answer your question? What was? Okay. I didn't make it sure. Because sometimes I get on.
The bacteria cells will be able to fit inside.
Yeah.
Fit inside what?
So the bacteria cell is smaller than the cells in your body. So the bacteria cell can get inside the cells in your body. It's called an infection. This slide. All right. So all cells share common features. All right. So we have certain common features that are common to all cells. All right. So cells generally have genetic material, DNA. They have ribosome, cytoplasm, ways to make protein, cytoplasm that kind of holds everything together, or a space for all the different organelles to be existing in, and then we have what is called a cell membrane. And it explains these in the next coming slides. All cells are going to carry out chemical reactions needed to sustain life, such as cellular respiration and protein synthesis. And if you look at the human body, for example, there are millions of chemical reactions going on at any specific time in the body. All right? I look okay? Should I go on this side? Joking. All right. Why are cells so small? Great question. Smaller cells generally have more surface area relative to their volume. High surface area allows the cells to quickly exchange materials. Remember, even though cells are the smallest unit of life, they're interacting with whatever's around them. Right? So things like the oxygen that you breathe. Huh?
Sorry, I just delivered a question on the quiz, right?
Oh, okay, great. The oxygen that you breathe is traveling through your bloodstream, and then it has to get to the different cells in your body, right? Every cell in your body needs oxygen. Every cell that is living in your body needs oxygen. How does it get from the bloodstream to the cells that need oxygen? Well, it has to travel through the cell membrane, all right? And so cells generally are small, things travel through the surface, the cell membrane, as you see here, cell membrane travel through the surface, and things like oxygen can freely travel through the surface of each cell. That's one of the mastering concepts. We won't go over that right now. All right, so life is classified into 3 domains: prokaryotes, eukaryotes. We'll see the different domains in the next coming slides. So prokaryotes, the first one, they are small, simple in structure, and lack a nucleus. Okay? Bacteria, remember, bacteria can fit inside of a human cell. It's very small. Bacteria and archaea are 2 different domains of prokaryotes. All right? They lack a nucleus, small, simple in structure, and lack a nucleus. All right? This is under a high-powered microscope. Eukaryotes, larger, more complex human beings. Eukaryotes, larger, more complex with many internal parts, including a nucleus, that's the key element, nucleus, and other organelles, membranous organelles. Protists, fungi, plants, and animals are all eukaryotes. We have nucleus, we have membrane-bound organelles.
Here's a question.
Oh, sorry.
So I always hear about these eukaryotic cells. It's not eukaryotic, the eukaryotic cells.
Yeah, this is eukaryote.
Do we have multiple? Do we have multiple cells of those, or just one?
No. It's a class. So plants are eukaryotes. Humans are eukaryotes. Fungi, protists, they're all eukaryotes. We all have nucleuses. We all have membrane-bound organelles inside of our cells. That makes us eukaryotes. Huh?
That's how you like specify.
That's a classification of cells. So like, so there are some features that are common to all 3 domains. All right, so prokaryotic, no nucleus. The archaea prokaryotic, also no nucleus. Eukarya or eukaryotic has. Hello, class. And so continuing on with our chapter 3 and this cell. Right, so we have different types of cells. We talked about the eukaryotes and eukaryotic and the prokaryotic. Bacteria are prokaryotic. So as you see here in this diagram, they lack a membrane-bound organelles. That's what prokaryotes all lack. They lack membrane-bound organelles. The ribosomes in their DNA, remember DNA, deoxyribonucleic acid, that's our genetic information. But the DNA in the ribosomes in bacteria are free floating in the cytoplasm, as opposed to in eukaryotes, they are bound in a membrane-bound structure. So slightly different. And so we want to recognize those differences. As you see here in the animal cell, plant and animal cell, but here the animal cell are eukaryotic. And so they have different membrane-bound organelles. My daughter coming in. So it's not free floating. DNA is not free floating. Ribosomes are membrane bound. They have a membrane around them. They're not just free floating in the cell. So differences, a big difference between the eukaryotic and prokaryotic, between the animal and the bacteria, for instance, cell, for example. And these are things we need to, that help us classify cells. Different sizes, different shapes, membrane-bound organelles, non-membrane-bound organelles. Plant cells are also eukaryotic, and so therefore they have membrane-bound organelles. So they're different organelles, they're different the nucleus, the mitochondria, the lysosomes, all have a membrane around them inside the cell, right? They also have a large central vacuole and a cell wall. We see that in the plant cell, right? So slightly different structures in the plant cell. Eukaryotic cells divide the labor. Talk about that. And so we see here an animal cell and a plant cell, right? So the membrane, there's a membrane that surrounds each cell. So not only in eukaryotes do we have membrane bound organelles, so each organelle has its own membrane inside the cell, but the entire cell has a membrane around it, which regulates the passage of substances going in and out. They call it semi-permeable. So things like oxygen, you want that to be able to come into the cell. Things like water, you may want that to come into the cell, but certain things you may not want to come directly into the cell. Ideally, you would not want bacteria to just be able to pass through into the cell, right? So we need to have some means of regulating, controlling what comes into and out of the cell. And the membrane of the cell membrane helps with that, also helps maintain cell homeostasis, keep things regulated in the cell. That cell membrane is made up of what is called phospholipids, right? We talked about lipids as one of the organic compounds. And for the phospholipids, it's basically, you see on the side of the screen here, hydrophilic head, which is a phospholipid, a phosphate group, and a glycerol. And then you have these hydrophobic tails, which are these fatty acid chains, right? Very similar. That's the lipid. So the phospho is more at the top, and then the lipid is at the bottom. And what we have with membranes, cell membranes, is we have a phospholipid bilayer, right? So these phospholipids are amphiphilic, right? The top part, the phosphate group and the glycerol, is hydrophilic, means that it is attracted to water, versus the bottom part, if you remember, things like fats and oils are hydrophobic. So it's non-polar, and so it will repel water. Okay, so you have a hydrophilic head and a hydrophobic tail. And sorry, what did I do? Yes. And then we put that together. We put the tails together and the heads are separate, and that's what makes our phospholipid bilayer, which is selectively permeable to lipids and small nonpolar molecules. And that's what makes our cell, that's the base for our cell membrane. If you were thinking about sort of like a wall, this would be like the dry wall or the sheetrock of the wall. There's other parts and pieces to it, but this would be the main structure of the wall, or maybe it would be the studs of the wall, I guess. I don't know. So there are other things in the cell membrane besides just the phospholipid bilayer. There are membrane proteins, there's cholesterols, there's sugars, steroids, and other things that are inside that cell membrane surrounding the cell that is selectively permeable to purposefully allow certain things in and try not to allow other things into the cell membrane. And so we have proteins, enzymes, cholesterol, sugars, all part of the cell membrane. Chains of sugars, as you see here, and attached to the proteins and the phospholipids, play a role in cell-to-cell communication. We have those purple proteins that are embedded in the cell membrane will give you, they can be transport proteins, they can work with different enzymes. All different things are happening here in the cell membrane, but these all function in the homeostasis of the cell, right? The cell membrane is like a jello, they call it a fluid mosaic model. And so it's because you have these phospholipids, you have these proteins, you have these cholesterols, these sugar molecules, all these things involved in the cell membrane. These are all moving parts. So that it's a fluid type of thing. It's like a jello, but it's protecting the cell, protecting the inside of the cell, right? So, but after we get past the cell membrane protecting the cell, then we also have different organelles involved inside the cell. One of the most important parts of the inside of the cell is the nucleus. The nucleus of the cell contains the DNA, deoxyribonucleic acid, DNA, which is the recipe for proteins. And what am I saying there? The DNA of your cell is a code. So it's a chemical code. And what does it code for? It codes for different proteins that your cell and your body need. Proteins are the building blocks of our bodies, right? And so muscles are protein, bone has protein. There's proteins all over your body. And so your body needs to code for different types. You have transport proteins that are inside your cells, transporting things into and out of the cell. All of these proteins have different codes. The code for these different proteins is housed in your DNA. And so, and that is inside your nucleus inside your cell. Right? So, how do we get from DNA to protein? Well, that's a process. Right? So, the first thing that happens is messenger RNA, known as mRNA, matches the sequence of the DNA. So, you have DNA has the code for all the proteins in your body. It's a lot. We may only need one protein to be made right now. Like right this moment, this cell needs to make this specific protein. So we're going to copy the code for that specific protein, and we copy it into what is called messenger RNA, right? And then that messenger RNA can leave the nucleus through a nuclear pore, and go out of the nucleus. It's going to then meet with some ribosomes. When you see ribosomes, think of the area of protein synthesis. And so that nucleus, that messenger RNA, that mRNA, has now left the nucleus. It's bound to a ribosome where it can attach to the endoplasmic reticulum, which is a sort of pathway and packaging system inside the cell. And then what happens there is we have that ribosome and that mRNA can go through the process of figuring out the protein, which is made up. Remember, proteins are made up. The building blocks of proteins are amino acids. Figure out which amino acids we need to put together. Amino acids are the monomers for proteins. So what a protein is an amino acid chain that's been bound together and then folded a couple different times. And so the code for the amino acids is in the mRNA now. So we use at the ribosome, we can decode that mRNA code for each amino acid in that chain. And then that chain of amino acids, we call it a polypeptide chain, is going to fold on itself and make protein. So now we also have an endomembrane system. Once we make things inside the cells, we may need to move them around the cells, right? Think of this, if I make something, package it up, and it may need to go out of the cell. It may need to go to a different part of the cell. So we have this endomembrane system, which consists of the nuclear envelope, endoplasmic reticulum, also known as ER, your Golgi apparatus, lysosomes, vacuoles, and cell membrane. in the example here, in the mammary glands, as you know, from women who, when they are with child, their mammary glands will produce milk for the baby. Different parts of the endomembrane system will work together to secrete the protein that's in that milk and move them outside of the cell. Right? We have to make that protein that's in that milk for the baby. And it has to be, it's coded for in our DNA, copied by our mRNA, translated at the ribosome, and we make the chain of amino acids, which are folded, packaged, and now we have the protein, and then that needs to exit the cell so that the baby has the right protein in its milk. Right, so here the protein exits the rough endoplasmic reticulum and bubbles of membrane called transport vesicles. Right, so things that are going to exit the cell get packaged in a membrane. Remember, eukaryotes are membrane bound. And so things that are going to exit the cell get packaged in a membrane, a phospholipid bilayer membrane. The vesicles leaving the rough endoplasmic reticulum fuse with the Golgi apparatus, a stack of membrane sacs that acts as a processing center, right? So we form this vesicle, this membrane-bound protein. It goes to the Golgi apparatus, gets processed further. Proteins leaving the Golgi and transport vesicles, then fuse with the cell membrane. So the same type of membrane that's surrounding this protein that's been processed by the Golgi apparatus, the same type of membrane is the same type of membrane we have at our cell, the phospholipid bilayer. And so the two membranes can bind together and the protein can be secreted out of, it can bind together and then fuse with the plasma membrane, and then the packaging inside of the protein, the protein inside of the membrane bound protein, that the protein can be secreted out of the cell. Right. We also have cellular digestion. So some transport vesicles leaving the Golgi carry enzymes that catalyze hydrolysis reactions. So these vesicles will fuse with the lysosome. So think of it this way. There are some things in the cell that need to not be in the cell. You have to have a way to remove, not necessarily move things out of the cell like proteins that are needed for the mammary glands, but you need to also be able to, if something is no longer needed in the cell, or if there's debris inside the cell or waste inside the cell, or something that's not supposed to be inside the cell, we have to have a way to break it down, to digest it, to destroy it. And so we have vesicles packaged by the Golgi apparatus, make it another membrane-bound vesicle around this trash that's in the cell, and we send it to the lysosomes, which is a different organelle inside the cell. And that's where we have cellular digestion, to break down something that is no longer needed inside the cell. Break it down, and then it can be excreted outside the cell. That membrane can bind also to the plasma membrane and then open up and excrete it outside of the cell. Plant cells also have large central vacuoles, right? So a little bit different in plant cells. All right. So they don't have, they generally, plant cells don't have lysosomes, but they have vacuoles. And in these large vacuoles, they have cellular digestion, which can help regulate the size and water balance of plant cells. So you have to have a way to break things down, digest things, and the lysosomes in the animal cells or the vacuoles in the plant cells help with that. There are also peroxisomes, which can aid in digestion, very similar to lysosomes, but they can break down toxic substances inside of the cell. So they originate at the ER, the endoplasmic reticulum. Think of the endoplasmic reticulum as like a sort of like a transport mechanism or a highway inside the cell. And the peroxisomes contain enzymes that can digest and then oxidize certain toxic molecules. So when you do things like drink alcohol, your peroxisomes can help digest that toxic molecule that you're putting into your body and make sure that it doesn't stay inside of your cells. There's also energy related organelles. You may have heard of this one, mitochondria. So almost all eukaryotic cells have mitochondria, which extract energy from food, right? So we eat food, it's digested in our small intestines, brought to our cells. Our cells have to take that food and use and extract the energy from that food, break it down and extract the energy from that food and convert it to an energy that the cell can use so that the cell can do the different chemical reactions and the different functions necessary for you to stay alive. So cellular respiration converts sugar energy into a form the cell can use for work, and that happens at the mitochondria. Mitochondria. The space inside the mitochondria is called the matrix. Right? There's different folds in the inner mitochondrial membrane called cristae, and these are sites for chemical reactions. This is the powerhouse of the cell, or like the almost like the gas station of the cell. This is going to produce the energy that we need for the cell to be able to do different functions. All of these things that are happening, like producing proteins, moving things into and out of the cell, all that requires energy. Mitochondria provide energy for that. Energy can come from sunlight, as you know, if you have plants, maybe you have some plants in your house, and they don't eat food per se, but they can get energy from the sun. So eukaryotes that carry out photosynthesis include plants and some protists, right? So they have chloroplasts, these organelles. They're different from mitochondria, but they can convert energy from sunlight into sugar, which is food for the cell. Now, wouldn't that be wonderful if we could do that as human beings? You may think, oh man, that would be great. Wow, if human beings had the ability to just create energy from the sun. But then what would you wouldn't have a need to eat that cheesesteak and that you know those fries and all of that stuff. You wouldn't be able to take, you wouldn't have a need to taste all that good food. So would it be nice if we could get energy from the sun? It would be nice in a sense that nobody would starve, but it wouldn't be nice because we love to eat. Eating is more than just getting energy for most of us. It's enjoyment, it's socialization, it's all those good things. I remember my dad saying once upon a time, he said, one day we're gonna be able to just take a pill and it'll be a whole meal in a pill. And I said, Dad, it'll never happen because people love eating too much. God rest his soul, but he never saw that. He didn't live to see that happen, and I don't think I will either. Also imagine if you sit out in the sun too long and you got fat because you sat out in the sun too long. If we had chloroplasts in our bodies, we might get fat from sitting out in the sun too long. That wouldn't be good. But yes, we do not have chloroplasts, and we have mitochondria. But plants do have chloroplasts. They can convert energy from the sun into sugar, which they can use for food. So photosynthesis takes place inside of the chloroplast. They have a space inside called the stroma. Also have flattened sacs of internal membranes called thylakoids. And this is their site for the chemical reaction, photosynthesis. And then the sugar then travels to mitochondria, which extract the energy used for the cellular process. Did I say? I didn't. They do have mitochondria, but they get energy through the chloroplast. They don't have to eat. That's what I meant to say. I think I said it wrong. That's what I meant to say. Then we have also the cytoskeleton. Cyto. When you see the prefix cyto, that means cell. Obviously, skeleton, structure that supports us and gives us the ability to be able to stand up, and it's where our muscles pull on and gives us infrastructure. Right, so the cytoskeleton, the skeleton of the cell, is a network of protein tracts and tubules found in eukaryotic cells. And they have different functions: structural support, obviously, skeleton, cell division, transport, and cell movement. So you have different types of parts of your cytoskeleton: microfilaments, intermediate filaments, and microtubules. All right, so different types of cytoskeleton. The microfilaments help with the cell contraction and the change in shape. Intermediate filaments provide the strong scaffold, mechanical strength, and the microtubules act as trackways for moving organelles and vesicles. And so they each just have different functions, different components of your cytoskeleton have different functions. You may have seen cilia and flagella. Maybe you've seen sperm at one point in time in under a microscope or on a video, which is pretty cool to see. But microtubules make up cilia and flagella, and they are very important when you are swimming or moving. Sperm use flagella to swim. Okay. Also, very important in the cilia are very important in our airways to be able to push. So if you breathe something in and dust in, it helps push particles out of your airways. My daughter has an issue with her cilia in her airways, and so that's why her body can't move mucus and dust normally. Cilia dyskinesis, and so sounds kind of weird, but just the function of the cilia is off, and so it doesn't push dust and mucus away like it's supposed to. But yes, these are ways the cell can move, different ways it can move. Cilia is one way, and flagella allows sperm cells, for instance, to swim. So all made out of microtubules. You also have motor proteins. So inside cilia and flagella, microtubules slide against each other with the help of a motor protein called dynein. And the cilium or flagellum can bend and move, and you can get movement. Plant cells are surrounded by a wall. Main components of a plant cell are cellulose and lignin. Right? So the cell wall. So you don't see this, you don't see the cell wall. You see in animal cells, you see a cell membrane, but in plant cells, you see a cell wall. The cell wall gives the plant cell strength, regulates the volume, prevents it from bursting from water, and also roles in cell specialization. Oops. Plant cells communicate through plasmodesmata, channels that pass through the plant cell wall. So cell-to-cell communication is also important in animal cells, but plant cells, they have this way that plant cells can communicate with other plant cells. Nutrients, biochemicals pass through channels to adjacent cells. So communicate. Um, it's pass nutrients through from cell to cell, right? So different ways the cells are connected and can pass things. Cells in multicellular organisms must constantly communicate with each other for tissues to function. Remember, cell is the basic building block of life, the smallest unit of life. Cells working to different types of cells working together make up what is called a tissue. Tissues working, different tissue types working together make up what is called an organ. Multiple organs working together make up an organ system. And then all the organ systems in your body make up what is called an organism. Right, so keep that in mind. But all of these cells need to communicate with each other, whether you're on the tissue level, the organ level, or the organ system level, or the organism level. Cells have to be in constant communication. I need to know what we need to be working together as an one big organism, right? So proteins and carbohydrates protruding from the cells are crucial for, helping to recognize cells, to recognize other cells, and to be able to communicate cell to cell. Animal cells have different ways that they're connected to each other to be able to also, similar to plant cells, to be able to also communicate. Gap junctions in animal cells are very similar to the plasmodesmata in the plant cells. These gap junctions are almost like a tunnel from cell to cell, and allow things to move between cells. Cells can also be bound by tight junctions, which are tightly bound the cell, but they generally do not allow things to move between the cell, and allow the cells to be connected tightly so that things don't leak between the cells. You also have anchoring, adhering junctions, connect cells to other cells and to the extracellular matrix. And not all cells are equal. Muscle cells can contract. Leaf cells have chloroplasts, so they carry out photosynthesis. Cells can be very different from each other. A lot of, when we talk about cells, the anatomy, the structure of the cell has a lot to do with the physiology, the function of the cell. And so some cells are big, some cells are small, some cells are long, some cells are short, some cells are round, depending on what they are, what their function is. And so we'll continue to put this together with more information next time. But the cell is amazing. A single cell. We all started off as a single cell. And look at you now. Goodbye for now.